chose to recreate DNA synthesis using Play-Doh, which is a substance that can be modeled into any shape and size that is wanted. We were not bounded by definite shapes and structures.
This helped us as a group to better understand DNA synthesis because it was not a flat picture. It was a 3D model that we ourselves could move and play around with to see the different possibilities of movement.
The significance of DNA synthesis is before a cell can reproduce, it has to replicate the DNA so that there is enough for more than one cell. DNA resembles a twisted ladder. It is referred to as a double helix. The bases of the ladder are sugar phosphate backbones. The rungs of the ladder consist of the nucleotides adenine, guanine, cytosine, and thymine. The A and T nucleotides always pair together, as well as the C and G nucleotides. Nucleotide pairs are held together by hydrogen bonds. The bases of the ladder are antiparallel, each having a 3' end and a 5' end. Synthesis begins with the enzyme. The helicase attaches to the DNA and moves along its length, unzipping and unwinding the DNA as it goes. As it unzips, the replication fork forms. As the helicase unzips the DNA, it puts the double helix under a lot of pressure. Topoisomerase attaches to the parental strand ahead and the replication fork, keeping the DNA from breaking or cracking. Next, single-strand binding proteins bind to the separated parental strands, keeping them from annealing or reattaching. Next, the enzyme primase comes along and attaches to each of the template strands. Each primase lays down an RNA primer. These are complementary strands of RNA paired with the appropriate DNA nucleotide. After the primers have been attached to the template strands, new nucleotides can now be attached. DNA polymerase III attaches to the 3' end of the RNA primer and begins adding new nucleotides to the parental strand in the 5'3" direction. DNA continues to elongate with the help of DNA polymerase III, but because of the antiparallel directionality and the fact that DNA can only build 5'3", a problem arises. Both template strands cannot elongate in the same direction. The top strand, the leading strand, elongates towards the replication fork with no interruptions, but because of the 5'3" rule, the lagging strand must build away from the replication fork. The lagging strand must elongate discontinuously, with a new RNA primer being laid at the beginning of each fragment. These are called Okazaki fragments. Once elongation has completed, DNA polymerase I must replace the RNA primers with appropriate DNA nucleotides. Now we are left with breaks in the new sugar phosphate backbone, where the start and finish of the RNA primers once were. The enzyme ligase comes along and repairs these breaks, filling them in with phosphodiester bonds. We are now left with two complete DNA structures.